Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about in situ hybridization, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss indicative repertoire, knowledge requirements, competence requirements, reflective statement, evidence of achievement, Q&A suggestions, evidence ideas, and some helpful tips to make candidates' portfolio really good. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into the first subsection of the fourth section, molecular testing, called in situ hybridization. The portfolio offers indicative repertoire for this section that includes HER2, HPV, EML4 ALK fusions, EBV, immunoglobulin mRNA. The knowledge requirement explains what knowledge is expected of the candidate in relation to the relevant subsection. Candidates should be able to demonstrate their knowledge at the specialist level. Think about what knowledge they had as a Band 5 BMS. How would that differ to the knowledge of a Band 6 BMS? That specialist level of knowledge would allow the candidate to troubleshoot, problem solve, verify or validate. The first knowledge requirement is principles of in situ hybridization techniques, including the use of fluorescence and chromogenic methodologies. Candidate needs to have the knowledge and understanding about in situ hybridization. You could ask the candidate to explain, what is in situ hybridization, to name steps taken during this technique, to explain why is this technique used in histology, to list differences between fluorescence and chromogenic method, to name their advantages and disadvantages. When addressing this knowledge requirement, candidates should consider sample preparation, fixation, permeability and pretreatment, denaturation, hybridization, washing, and visualization, evaluation, advantages, disadvantages, and application of FISH versus SISH methods. The second knowledge requirement is role of the clinical application of in situ hybridization in diagnostic pathology. Candidate needs to have the knowledge and understanding about application of in situ hybridization. You could ask the candidate to explain how the in situ hybridization is used in diagnostic pathology, to list the clinical applications of in situ hybridization, to highlight the limitations and challenges with in situ hybridization. When addressing this knowledge requirement, candidates should consider role in oncology and infectious diseases, pathogen identification, detection of gene expression, amplification, rearrangement, translocation, fusion, disease monitoring or guiding therapy, the limitations and challenges of in situ hybridization. Competence requirement is basically a combination of knowledge, skills, and experience candidate needs to perform a job to a satisfactory standard. Candidates' chosen evidence should demonstrate these competence requirements. The following competence requirements need to be met for this subsection. The first competence requirement is prepare cell and formalin fixed, paraffin wax embedded samples for DNA and RNA analysis. Candidates should be able to prepare samples for DNA and RNA analysis. Does the candidate prepare cells and FFPE samples for DNA and RNA analysis? Take correct steps and understand why they take them? Do they follow local protocols and SOPs? Do they correctly troubleshoot problems encountered during DNA and RNA analysis? When addressing this competence requirement, candidate could consider deparaffinization, rehydration if needed, permeabilization, denaturation, hybridization, detection, visualization, SOPs, method used like FISH or SISH, your training and competence. The second competence requirement is stain tissue sections using in situ hybridization. Candidates should be able to stain sections using in situ hybridization. Does the candidate stain tissue section using in situ hybridization? Do they take correct steps and understand why they take them? Can they troubleshoot problems encountered in situ hybridization? Do they follow local protocols and SOPs? When addressing this competence requirement, candidate could consider tissue preparation prior staining, stains highlighted in indicative repertoire and stains used in your department, SOPs, methodology used, control material, your training and competence. The third competence requirement is select control material, maintaining appropriate audit trails. Candidate should be able to select and use correct control material. Does the candidate use correct control material? Do they maintain correct audit trail for control material used? When addressing this competence requirement, candidate could consider tissue types, verification, validation process, record keeping, traceability, audit trail, importance of selecting correct control material, consequences of not using correct controls. The fourth competence requirement is use appropriate microscopy techniques to visualize stained material. Candidate should be able to microscopically assess stained slides. Does the candidate use appropriate visualization techniques to assess stained slides? Do they use light microscopy, immunofluorescence, and filters during the slide assessment? Do they set up microscope correctly? When addressing this competence requirement, candidate could consider light microscopy, immunofluorescence, filters used, magnification, Kohler's illumination, digital pathology, and AI algorithms. The fifth competence requirement is assess quality in prepared sections. Candidate should be able to assess quality of stained sections. Does the candidate assess stained section for quality correctly? Do they take correct steps during the assessment? Do they focus on correct features when assessing quality of staining? Do they maintain appropriate audit trail? When addressing this competence requirement, candidate could consider staining intensity and uniformity, specificity and sensitivity, presence of background, consistency of staining, presence of nonspecific staining, signal intensity, EQA assessment criteria, relevant documentation, control material. The sixth competence requirement is clearly distinguish between positive, negative, and equivocal results. Candidate should be able to distinguish between positive, negative, and equivocal results. Does the candidate distinguish between the positive, negative, and equivocal results? Do they follow the correct procedures? When addressing this competence requirement, candidate could consider SOPs, methodology used, their training and competence, controls used, ratio, copy number, signal, characteristics, criteria, interpretation. The seventh competence requirement is resolve problems associated with the demonstration methods, including root cause analysis, as appropriate. Candidate should be able to troubleshoot staining. Does the candidate resolve problems with staining correctly? Can they identify and solve the common problems? Do they record problem solutions accurately? Can they perform root cause analysis? When addressing this competence requirement, candidate could consider investigation, problem identification, corrective actions to fix the issue, preventive actions to minimize problems from reoccurring, possible problems like poor staining intensity, poor specificity of staining, artifacts like uneven staining, hazy slides, presence of deposits or precipitate on the slide, reagent contamination, effects of fixation, processing and antigen retrieval on quality of IHC staining, nonspecific staining, background, absent staining, cross-reactivity. The eighth competence requirement is dispose of waste reagents and residual material safely. Candidate should be able to safely dispose waste reagents. Does the candidate dispose staining reagents and residual material safely? Do they follow local procedures and avoid risks associated with staining reagents? Do they segregate waste as per regulations? When addressing this competence requirement, candidate could consider licensed contractors used by your lab, waste segregation, safety data sheet of reagents that often state disposal ways, risk assessments, local policies, and regulations that are followed, health and safety risks associated with reagents like flammability, corrosivity, environmental toxicity, risks associated with disposal or residual material like human tissue. Each section also contains reflective statement which is asking the candidate to summarize their role in the lab in the context with each section. When they complete all the subsections within each section, they will be required to write a reflective statement. They also need to write a personal reflection on their training and why they chose their evidence. To prepare for that, ask them to write a smaller reflective statement for each subsection. What is their role and context of this subsection within the lab? What does their evidence demonstrate? Ask them to write a smaller justification as to why they chose their evidence for this subsection. Also ask them to write about what was their training like when to this subsection. Writing a smaller reflective statement at the end of each subsection will then make it much easier for them at the end of the section. Each subsection also has evidence of achievement. This is a part where you, the training officer, sign the candidate off as competent and fit to practice as a BMS at the specialist level. By doing that, you will confirm that the candidate can work in accordance with local SOPs relevant to this subsection. This will also confirm that candidate has answered questions set by you to test their knowledge and skills relevant to this subsection. And you will also confirm that candidate's chosen evidence demonstrates their competence, relevant to this subsection. The candidate will need to be signed off as competent by an internal assessor for each subsection. They can be signed off by you, the training officer, senior BMS overseeing relevant section, or by the training lead. These are some example questions you could give your candidate to test their knowledge and skills about this subsection. These should be based around the knowledge and skill requirements mentioned earlier. You could ask the candidate to complete tables you created, based on knowledge requirements. Supporting evidence should be selected by the candidate. It needs to be based around the competency requirements, and be relevant for each subsection. The evidence should allow the candidate to demonstrate their competence in each subsection. It should be specific and relevant to the candidate's in-house practice. It should also demonstrate candidate's specialist competence. If you would like to see specific ideas for supportive evidence, please have a look at the video for trainees. Link will be included in the video description. I would also like to share with you some helpful tips that can make candidates' portfolio look really good. Ask them to try and present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs, or even videos, if they can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Ask them to try and make it a little bit more interesting to the reader, which will be you, and eventually the assessor. Ask them to try and put themselves in your or the assessor's shoes. How would they like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture 1,000 words. I will make a video where I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also ask them to try and use various types of evidence, not just one type, like case studies, for each subsection. They could use experiments, case studies, annotated results, annotated photographs of them performing tasks, evidence of troubleshooting, annotated EQA results, nonconformances, validation verification evidence, comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Ask them to try and bring in their personality. Ask them to think about the ways they like to learn best. If experimenting, let them perform experiments. If they like troubleshooting, let them perform troubleshooting. Ask them to choose something that demonstrates their specialist understanding, knowledge, and skills, and how they apply this in their practice. And one more thing, ask them to keep the competence requirements in mind when they are selecting their evidence. Good evidence will tick off several requirements. And this brings me to the end of today's video. Hopefully you now have a better understanding about the knowledge and competence requirements for this subsection. And I hope you also have a better idea what questions you can choose to test candidates' knowledge. And my final take-home message? Keep those requirements in mind when selecting questions and guiding the candidate. Bye for now.